Hello students, so we are going to learn about bacterial cell wall today. So this image shows us a typical profile of the bacterial cell envelopes. So gram-negative bacteria, they typically contain an inner membrane followed by a thin layer of peptidoglycan, which is present within the periplasmic space, followed by the outer membrane, that is lipopolysaccharide in nature. Additionally, you may also find a layer which is known as a capsule layer, which may or may not be present in some bacteria. Gram-positive bacteria, on the other hand, shows an inner membrane followed by a very thick peptidoglycan layer, which is basically multi-layered. Outer membrane is absent in case of gram-positive bacteria, and capsule may or may not be again present. So the primary function of the bacterial cell membranes is to prevent the rupture of the bacterial cell and to protect it against osmotic lysis. So inside the bacterial cell membranes is present the protoplast. Now this is just another image of the comparative image of gram-positive and gram-negative cell walls. This slide talks about the principle of gram staining method and how we can observe the different staining properties of gram-positive and gram-negative bacteria. So gram-positive bacteria, they basically have a multi-layered peptidoglycan and a very less lipid content, while gram-negative bacteria, they contain a very thin layer of peptidoglycan, and they contain more of lipid contents. So here, during gram staining, we make use of 4 reagents. The first is crystal violet. That is, after crystal violet staining, both gram-negative and positive are going to get stained purple in color. Then we do the iodine treatment, where we have formation of the crystal violet iodine complex on both gram-positive as well as negative. So here, again, both will appear purple in color. Followed by this, we'll have a decolorization step in which the gram-negative bacteria, which contains more of lipids, they get dissolved in the decolorizer and leads to formation of larger pores in the middle. While gram-positive bacteria that have less lipids and more peptidoglycan, they don't have formation of large pores in the membrane by dissolution of lipids, as the lipid content is as such less. So after this, we wash with water, during which the gram-negative bacteria that have large pores, they will lose the crystal violet iodine complex, while gram-positives will retain the complex, and hence after counter staining, with the second dye, that is safranin, the gram-negative will take up the second dye, will appear pink in color, while gram-positives will retain the primary color, which is of crystal violet. So this method is basically used for differentiation of gram nature of a bacteria, which is a part of biochemical reactions used for identification of bacteria. Speaking about the chemical nature of the cell wall, so the bacterial cell wall is basically made up of a peptidoglycan material that is basically made up of 2 sugars. So here you can see the structure of peptidoglycan. The 2 sugars that are present are N-acetyl glucosamine and N-acetyl uranic acid. So we'll talk about the detailed structure of these 2 sugars in the later part of the presentation. So cell wall material is also only present in case of bacterial membranes. So coming to the structure of peptidoglycan, it is basically made up of glucose and its derivatives. So D-glucose molecule, here we can see the structure of D-glucose, which has 6 carbons in the backbone and is an adenose sugar, and showing the CHO functional group. Now, while being present in biological systems, we don't have presence of linear form of the sugar, but they are present in cyclic forms. So for conversion of a linear structure into cyclic structure, we basically have addition of water molecules first across the double bond here. So water will split into H plus and OH minus and will get added across the double bond. So you have addition of 2 or presence of 2 hydroxyl groups at carbon number 1. This is followed by a loss of water molecule between carbon number 1 and carbon number 5. So loss of water molecule here will lead to formation of a cyclic ring structure. Okay. This cyclic ring structure of beta D glucose can also be represented in the form of a pyranose ring structure, as seen in this image here. Okay, so this is the cyclic structure of beta D glucose molecules. So the sugar can be alpha or beta sugar depending on the orientation around carbon number 1. So if the hydroxyl group is present above the plane of the ring, it is beta sugar. If it is present below the plane of the ring, it is alpha sugar. So here in case of peptidoglycan, we have presence of the beta sugar. Further, the first sugar in peptidoglycan is an acetyl glucose amine, that is basically derivative of glucose, which is formed by first addition of an amino group to the glucose residue at carbon number 2 here. So addition of an amino group first, and this amino group is further acetylated to form an acetyl glucose amine, which is the first sugar in peptidoglycan. The second sugar, which is for short known as NAM or N-acetylmuramic acid. So it is a further derivative of the sugar N-acetyl glucose amine. So further, we have addition of a lactic acid side chain at carbon number 3. Okay, so we have addition of a lactic acid side chain here. And further, to the carboxylic group of the lactic acid side chain, amino acid, short amino acid chain is added. As you can see here, there is a stretch of 4 amino acids which are added by a bond between the amino group of the amino acid and the carboxylic group of the lactic acid, forming a peptide group or a CONH bond. So this is the second sugar, that is N-acetylmuramic acid. So the function and structure of peptidoglycan, the function is basically to provide shape and structural support to the cell. It resists damage due to osmotic pressure. It provides some degree of resistance to diffusion of the molecules, and it is basically a single bag-like structure or covering that is present around the cell membrane. So here in peptidoglycan, these 2 sugars, that is N-acetylglucosamine and N-acetylmuramic acid, they are bonded to each other by beta-1,4 glycosidic bonds. So this one pair of NAG and NAM sugars is also known as one unit of peptidoglycan. So we have these alternating NAG and NAM residues present that will form long chains of peptidoglycan. And then remember, the peptidoglycan is basically a sac or a sheath that will surround the cell membrane or the inner membrane. So basically, to convert these linear chains of peptidoglycan into a sac or a covering, there needs to be formation of crosslinks between the individual chains. And these crosslinks are done by formation of bonds between the amino acid side chains. So these amino acid side chains are further bonded to each other by short interbridges which are again made up of some other amino acids. So this represents the diagrammatic representation of how the peptidoglycan structure is present in gram-negative bacteria. So you have linear chains of NAG and NAM, alternating NAG and NAMs that contain the amino acid side chains, and these amino acid side chains can bind to amino acid side chains of NAM residues present within the adjacent linear chains that are linked to each other by cross-links. So here in this diagram, we are trying to show that the peptidoglycan of bacteria are sensitive to antibiotic penicillin because it prevents formation of this interpeptide bond. And similarly, lysozyme is also can also target peptidoglycan by acting on the beta-1,4 glycosidic bonds that are present between the NAG and NAM residues. And hence, gram-negative or basically peptidoglycan molecule is sensitive to both antibiotic penicillin and enzyme lysozyme. So that's it, students. You can kindly like, share, and subscribe if you like the contents and get direct updates of the videos as and when I upload. Thank you.